all the elements of dopamine neurotransmission are in this very handy figure. So here's dopamine. Maybe these are all supposed to be vesicles filled with it. One of the first things to notice is this, the role of amphetamine, and as it turns out, cocaine, so we could use cocaine here as well, in dopamine neurotransmission. And the key there is what they do to the dopamine transporter. So both amphetamine and cocaine take the dopamine transporter, and instead of pumping dopamine back out of the cleft and into the cytosol, the transporter is reversed, and now it pumps dopamine out of the cytosol and into the cleft. And so you get this release of dopamine and its buildup in the synaptic cleft, all because the transporter's been reversed. That's the role. That's what amphetamines and cocaine do. You'll also notice here, postsynaptically, we've got drugs that work. Apomorphine and haloperidol. Apomorphine, it's an agonist, whereas haloperidol is an antagonist. In fact, there's a series of antagonists to dopamine, all of which are neuroleptics. And a neuroleptic is something that's used to treat affective disorders, particularly schizophrenia. In fact, years ago, Saul Snyder was able to demonstrate that, that's supposed to be an I, it's supposed to be a Z, that you can rank order the neuroleptics in terms of those that are most effective at lowest concentrations and those that are minimally effective even at high concentrations, and you find that that matches exactly their ability to antagonize dopamine binding to this receptor, the postsynaptic receptor. So haloperidol, a very effective dopamine antagonist used to treat schizophrenia. So far as breakdown of dopamine is concerned, you'll notice a couple of things. There are two enzymes that are going to be able to accomplish it. One is catechol-O-methyltransferase, and the other is monoamine oxidase. They convert dopamine to homovanillic acid. If you go, you can, each of these can work. That is, COMT can work first, and then monoamine oxidase secondly, or monoamine oxidase can work first, as happens inside the cell. That creates this thing called DOPAC, and then DOPAC gets worked on by COMT to produce homovanillic acid, HVA. All right, now what's important about that? DOPAC can be toxic. That is, if DOPAC builds up within the cell, it can be degraded by hydrogen peroxide and produce something that will actually attack the dopamine vesicles. That gets into the transporter of the vesicle, causes the vesicle to break apart. So using dopamine, using monoamine oxidase inhibitors, MAOIs, is something that's done in Parkinson's disease, in treatment of Parkinson's disease, for example. MAOIs are also used just to build up the concentration of dopamine, because without MAOIs, dopamine concentration builds up. That means you got more dopamine that can be released. That means there's more that hangs around in the cleft. MAOIs are most commonly used to treat depression. So they're antidepressants. Antidepressants. Okay, so those are the major elements here that are present that we have to worry about in terms of dopamine pharmacology. Cocaine and amphetamine blocking reuptake, haloperidol an antagonist, apomorphine a very nice agonist, and then the breakdown of dopamine by monoamine oxidase and catechol-methyltransferase. And there are two types of MAO, that which is outside the cell in the cytosol, and that which is inside the cell, inside the cytosol.